Well, good morning. I think that we are having folks join us now. I can see the participant count going up there at the bottom of my screen. And I just want to welcome you all here to today's entry into our series of distinguished lectures for the supporting the bioeconomy. This is the first panel session that we're having in this bioeconomy lecture series, and it is being presented by the whole series is being presented through our bioeconomy coordinating committee here within NSF and all of the NSF directorates. We've instituted this lecture series to highlight NSF's support for biotechnology that advances the bioeconomy as part of industries of the future or industries of tomorrow. The industries of tomorrow are cross-cutting, convergent, and interdependent fields of research that collectively offer enormous economic potential and are critical to the nation's long-term economic and national security. And certainly biotechnology can be described as the research, infrastructure, and workforce capacity that enable the discovery, utilization, and alteration of living organisms, their constituent components, and their biologically related processes. NSF's advancements to advance biotechnology serve to accelerate scientific discovery and to enable the application of that technology to create goods and services, or the bioeconomy, that contribute to the agriculture, health, manufacturing, and energy sectors, among others. So our speakers today, who are co-sponsored by the Bioeconomy Coordinating Committee, the Directorate for Biological Sciences, and the Directorate for Engineering, are some of the rising stars in the fields of biology, biotechnology, and synthetic biology. All four of our speakers today, I'm happy to say, have a long history of NSF funding across a number of programs within bio and engineering. Several of them also have funding from other directorates, which really truly speaks to the reach and the relevance of biotechnology across all the major disciplines. Several are also recipients of NSF's prestigious Career Award. Congratulations to all of you. The interdisciplinary nature of their work comes at the intersection of biology, biotechnology, and biodesign automation, and has the potential to advance new technologies to better prepare us for unexpected events, such as those we have seen with COVID-19. So not only are our four speakers invested in propelling the bioeconomy, but they hold a strong commitment to increasing the pipeline of students in the synthetic biology workforce. Today, in addition to their scientific achievements, they'll be presenting on their ongoing research efforts. So each is going to be introduced by a program officer who has funded their work. But let me just start by welcoming, and ask you to join me in welcoming Drs. Jennifer Nemhauser, Lydia Contreras, Julius, and Douglas Densmore to our virtual NSF. And I am truly looking forward to this panel. So let me turn it over, and I'm not sure who I'm turning it over to first.
I'm handling it, Anne
Excellent.
Sylvester.
Excellent. So Anne, please take it away.
Thank you, Joanne. I'm very pleased to introduce Dr. Jennifer Nemhauser today. Dr. Nemhauser is an HHMI scholar and a professor of biology at the University of Washington. She received her PhD in plant biology from the University of California, Berkeley, and went on to a postdoc at the Salk Institute for Biological Studies. It was during her time at the Salk Institute that she was first introduced to genomics, and she began to use genomic tools to further the understanding of plant hormone signaling. Jennifer's work is highly integrative, and it draws upon molecular genetics, genomics, physiology, and synthetic biology in order to build new tools to study plant signaling dynamics and to evaluate the impact of evolution on signaling networks. Today, Jennifer will give us an overview of how plants contribute to the bioeconomy and where specifically plant synthetic biology fits into this picture. She'll also illustrate her efforts in recruiting and retaining diverse teams of scientists, and she'll describe how she trains investigators to foster an inclusive climate and culture in science. Thank you for joining us, Jennifer.
Thank you so much. I think now there will be a pre-recorded talk coming up. Great, thank you.
Thank you. It is my great privilege today to talk about some of my favorite topics: plant biology, synthetic biology, mentorship, and art, and how they all can come together to advance the security and health of the economy. It's a lot of ground to cover, and we have some really great other talks that I'm looking forward to hearing myself, so I'm going to launch right into it. Plants are a key to any discussion of the bioeconomy. They are robust and able to survive in diverse habitats that are in a constant state of flux. They are plastic and can be readily adapted to meet human design specifications. They produce a range of amazing chemicals, including biomaterials that allow enormous growth and durability, as well as the potential to act as long-term carbon sinks. The Plant Science Research Network, which led to the development of the recently published Decadal Vision, is an NSF-funded RCN with representatives from 15 scientific societies. In 2019, which I have to admit feels like a lifetime ago, I was among the lucky 50 plant scientists who came together at Biosphere 2 in Arizona to discuss the future of research, training, and discoveries in plant sciences. The resultant Plant Science Decadal Vision has a central theme, which is that research, people, and technology must all be advanced for plant science to succeed and to grow. More than ever before, the future is about plants and how they can contribute to a sustainable future for humans and indeed for all life on the planet. The document is packed with specifics around 8 hubs, and I will draw your attention specifically to Hub 2, Advancing Technology for Diversity-Driven Sustainable Plant Production Systems. In the Decadal Vision report, we propose 2 far-reaching research goals or moonshots in this domain that I just want to present here as to give you a flavor of the way that we are thinking about these goals. The first is transforming cropping systems with new perennials and polycultures. So this is really thinking about what particular species we are growing, and the conditions in which we are growing them. The second moonshot was entitled Breaking the Water Habit by Building Resilient Crops for the Future. And this goal is really about changing the wiring within the plants to be able to allow them to meet current and future climate conditions. More than a quarter of the world population is currently experiencing moderate to severe levels of food insecurity. This incalculable loss to human potential will only grow more dire with the impending failures of the agricultural system, especially among populations that are already the most vulnerable. Solutions will have to come in many forms, and I strongly believe that technology, and in particular synthetic biology, must be among them. In particular, a new generation of smart crops engineered with modular solutions that are able to work across a broad range of plant species could allow local communities to gain control over their own destinies, increase the acceptance of engineered crops, and ultimately reduce human suffering. The economic impacts from this kind of paradigm shift will ripple out from direct impacts on the environment to improving human health to broader international concerns. So I mentioned synthetic biology, and I want to go deeper into that aspect since it's our topic for today. So in 2018, I was fortunate to participate in a Banbury conference with a very ambitious title, Revolutionizing Agriculture with Synthetic Biology. And the paper I'm citing here is one of the outputs. Although I wasn't an author of this paper, I really like how they're thinking about the problem. In particular, I love this quote. Many plant scientists, and I would argue many people outside of plant science, fail to recognize the vast scale of the potential design space for everything from metabolic enzymes to plant architecture, and how little of this space is occupied by modern plants. In this really great figure on the left of the slide, you can see the enormous number of creative concepts, and this represents just a fraction of what is possible and includes some of the topics that I highlighted from the decadal vision. And it also brings to the forefront some of the limits that we face. And I think one of the important parts to recognize here is that same theme of research, technology, and people. These limits come through in a different way here. To really overcome a lot of what are current bottlenecks, we will need innovations in training and new kinds of collaborations in addition to new technologies. So beyond the people that are within the education and research pipeline, there are other stakeholders that historically have been marginalized, excluded, or disrespected. These include the public on whose behalf we are working, farmers working in an enormous range of socioeconomic and environmental conditions, Indigenous communities with deep knowledge of specific plants and practices, agronomists, and decision makers in governmental and non-governmental organizations, just to name a few. How would our science be different if we were regularly engaging with these audiences, not to educate them, but to listen, to learn, and to build long-term relationships based on trust? There is clearly a need for funding and leadership to foster these type of exchanges. I would argue that one of the most effective ways to minimize harm with technology is to transfer expertise to scientists who live in the communities where new varieties are needed, allowing for local conversations about values and priorities to guide the decision of which products are developed. This can also extend domestically to some of the amazing organizations involved in connecting farming to social justice. Here in Seattle, we have groups like Black Star Farmers and Indigenous Roots as two examples. Okay, so now I would like to transition to telling you a little bit more about what my lab does in this domain. And before I get there, I want to briefly orient everyone to the way that I conceptualize synthetic biology, because I know it is actually, there are many versions out there. Probably every synthetic biologist may have their own. So for me, the formative document that really helped me understand what I wanted to do in my own lab was published in 2002 by Professor Hiroaki Kitano. And in this article, Kitano proposed 4 domains of inquiry that would lead to what he called a continuous spectrum of knowledge, from molecular, biochemical, and biophysical details up through organism-level behaviors. So in this figure, I've tried to represent these 4 different domains of inquiry. So structures represent experimentally validated connectivity maps of biological processes and pathways. Dynamics capture quantitatively how network behaviors change over time, often in response to particular stimuli. Control uses techniques like sensitivity analysis to identify which nodes in the network are best suited for engineering desired changes in network performance, i.e. phenotypes. And finally, design synthesizes the knowledge arising from the other 3 domains to modify and construct novel biological systems with user-defined behaviors. The intervening decades have seen remarkable achievements in the first 2 of these aims, largely under the umbrella of systems biology, but the second 2 have been more of a challenge, and I think synthetic biology has in many ways emerged as a field to fill this gap. My lab focuses specifically on the subdomain within synthetic biology of synthetic signaling, and we're particularly excited about understanding and engineering signal processing modules, what I often shorthand as plant logic. In studying plant logic, we can surface principles that underlie existing biological systems, as well as being able to engineer new systems. Our first big breakthrough in this space came as part of an NSF-funded collaboration with Professor Eric Clavens. It was a recapitulation of a simple pathway from plants into yeast. Namely, by porting 5 parts from the model plant Arabidopsis, we were able to trigger reporter gene expression in the presence of a small molecule hormone called auxin. The 5 parts are a promoter, the transcription factor that binds to that promoter, an adapter protein fused to a co-repressor that keeps the transcription factor inactive until a small molecule hormone glues the adapter to the receptor that then targets it for degradation. This degradation of the repressing element allows the transcription factor to activate transcription. This approach allowed us to tease apart the functions of different functional modules within the pathway, degradation, repression, activation, as well as to quantify functional diversification within the paralogs or isoforms of each component. So what did we learn with this technology? For starters, we were able to use time course flow cytometry to acquire the high-resolution dynamic data we needed to build simple models that enabled sensitivity analysis to find tuning knobs for different performance metrics. It also allowed us to start connecting natural and engineered variation in these tuning knobs to different developmental phenotypes. For example, we've mined the 1001 Genome Initiative to find receptor variants that alter root architecture. Recently, we successfully recapitulated the auxin response from maize into yeast as part of a PGRP-funded collaborative project. And our comparisons with Arabidopsis and maize have opened up our thinking in multiple new directions. So what did we build with what we learned? So one example on this side is that we have used the knowledge gained from parts characterization to build new technologies like the hackers shown here, which are synthetic hormone-responsive transcription factors, which rely on a catalytically inactive Cas9 fused to sequences that are necessary and sufficient for hormone-induced degradation and transcriptional repression. As a proof of principle application for this approach for engineering yield-associated properties, we directed an auxin-regulated hacker at a target gene, and by doing this, we were able to tune the level of feedback and also alter, and thereby alter, the number of branches in the plants. We're currently working on applying the same technology to understand why elevated CO2 levels are breaking many of the key yield-enhancing phenotypes in green revolution varieties, where another hormone, gibberellin, is the one that has been targeted. So as a means of transition, I want to share this incredible quote from a paper from Joe Thornton, someone who has been enormously influential in the way I think about protein design space and evolution. In particular, I want to share the very last part of the quote. The singularity of the present seems to rationalize the past. History leaves no trace of the many roads it did not take, or of the possibility that evolution turned out as it did for no good reason at all. I spend a lot of time thinking about systems and engineering systems, and as the plant decadal vision emphasizes, advancement is only possible with an integrated approach to development of research, technology, and people. Another way to ask this question is, how do we change the human-created systems in which our research is happening? In a recent podcast interview, Anna DeVere Smith was asked for her philosophy of teaching. She said that she tells her students, "Confidence is overrated. Give doubt a try." She went on to explain that doubt and humility are necessary preconditions for creativity. Many, if not most of us on this call, found a way to survive and flourish in the STEM research ecosystem as it currently exists. And it is not hard to look around and see phenomenal examples of brilliantly innovative science and engineering. This can lead to a false conclusion that the system is mostly working. It is not. And some of these headlines are just a very small number of ones that indicate why it's not. And the lost potential is impossible to quantify. So I want to spend the rest of my talk sharing two approaches that I'm involved in, which I hope might be making some headway in this area. The first is a joint initiative between the North American Arabidopsis Steering Committee, of which I am an elected member, and the American Society of Plant Biology. It's called Changing Cultures and Climates. There are several parts of this effort, all available and open to anyone who's interested through the Plantaid website. But I'm going to draw your attention to just a few right now. First, there is Front and Center, an initiative to highlight Black, Indigenous, and Latin plant scientists, and Adventitious Roots, a Discord server hosting private conversations among BIPOC plant scientists. These two efforts came directly out of conversations with a long-term cohort of NSF-supported BIPOC plant scientists, where we really asked them what they needed from their societies to feel supported. There's also a collection of resources gathered in an anti-racist toolkit with opportunities to share how the community is using these resources in lab meetings or departmental events or other activities. And finally, as part of this initiative, we updated the Diversify PlantSide database to make it more useful for anyone looking to expand their network of potential reviewers, speakers, mentors, grant panelists, etc. So in this last section, very briefly, I want to push this question farther into how we change our institutions. In this part of my work, I've been heavily influenced by art, particularly the field of institutional critique. Andrea Fraser, one of the founders of this field, beautifully reframed the question of how do we fix our broken institutions. She argued, every time we speak of the institution as other than us, we disavow our role in the creation and perpetuation of its conditions. We avoid responsibility for or action against the everyday complicities, compromises, and censorship, above all self-censorship, which are driven by our own interests in the field and the benefits we derive from it. It's not a question of being against the institution. We are the institution. It's a question of what kind of institution we are, what kind of values we institutionalize, what forms of practice we reward, and what kind of rewards we aspire to. In trying to think about my own practices, values, and reward structures, I have invited artists to join our work in a number of ways to help me gain this outsider's perspective that often helps one see hard to see things much more clearly. I've hosted artist residencies in my lab, followed by public reflections. I have brought artists into my classroom, and I have to say these ethics, values, and strategies are amazing. I particularly love the spaces of not knowing and practicing failing together. My lab has participated in art festivals, and I've even collaborated on a conceptual art piece that involved close collaboration with the Seattle Art Museum. Please feel free to ask me questions about any of these activities. I know they went by a little fast. When I imagine the next green revolution, I think about our goals, our approaches, and the people who will be part of it. I am incredibly fortunate to have a great team of people now and in the past years in my group and as our collaborators. And of course, I'm also grateful for my funding, including, as I hope I've highlighted throughout, the funding support I have gotten from NSF. So with that, I thank you all for your time, and I would be happy to answer any questions.
Great, thank you, Jennifer, for that wonderful talk. So we have a few minutes for questions for Dr. Nemhauser. If you would like to ask them, please insert them into the Q&A box. I will start off with a question about inclusiveness at NSF. So your passion for improving the inclusiveness of our institution really came through in your presentation. And if you had one piece of advice to offer us at NSF for improving inclusiveness, what would it be?
Yeah, thank you so much for the question. So I would say that one of the things I have been working on personally the most and have gained the most from is this idea of asking the groups that you are hoping to feel more included, what it is specifically that they need, and then doing that work. So instead of me deciding on the list, the punch list of things that I think are the most urgent things that are needed to be done, to actually ask in a genuine way and in a way that you can get an honest answer, so in a safe and in a place that encourages really frank conversation, what it is that is needed to change. And then regardless of whether that fits my own intuition, that I actually then do the work myself of implementing that with frequent interaction from the people who I'm hoping to help, to make sure to sort of continue the cycle of getting feedback and readjusting as needed to make sure that we're still getting towards the goal. I say in my lab a lot, like, you got to start with your goal and only then design process. I think we often get lost in process, especially as experimentalists, and then we can get very far away from the goal that we actually set, or don't even know what our goal is.
Great, thank you. The next question is, how do you communicate the value of your work to the public who may be reluctant to accept GMO foods?
Yeah, so definitely living in Seattle, I can say, I'm a regular attendee at the farmers market, or was before this last year. And almost all of the people that I interact with who are not scientists, even some of the ecologists in my department, I would say, are hesitant or outright hostile towards GMO technologies. And I think part of trying to figure out how to have those conversations differently is actually about trying to learn to listen and to really hear what the concerns are, and be able to take in that information and think about how to build trust and conversation. So rather than try to convince them, I try to really figure out what are the concerns and how can we adjust. And part of the work that I showed, like at the Arts Festival, was really amazing, because we put ourselves in this very vulnerable situation of going into a position where we as plant scientists were there, sort of showing ourselves as part of the community and just kind of answering questions about plant science and finding, like, really building rapport in a way that was that led to some fascinating conversations where you could start talking about some of the real issues in agriculture, about subsistence farming, about helping vulnerable populations, like shared goals and values, and then start talking about some of the ways that technology may or may not be the right way to approach some of those things. And I think that sort of doubt and humility part from Anna Devere Smith, I really took to heart. I think that is something that, as scientists, or I will just say personally, for me, I guess I sometimes find hard. I feel like training kind of pushed me out of being able to express doubt and humility in a lot of contexts. And it's actually exactly those skills that are needed, I think, to really talk to the public and to other stakeholders.
Great, thank you. So I'm going to hand this off to David Rockcliffe now, who will be introducing our next speaker.
Good morning, everyone. It is my great pleasure this morning to introduce to you Dr. Lydia Contreras. Dr. Contreras is an associate professor of chemical engineering at the University of Texas at Austin. She received her PhD in chemical and biomolecular engineering from Cornell in 2008 and was previously a postdoctoral fellow at the National Institutes of Health and also a visiting postdoctoral fellow at the Max F. Perutz Laboratories in Vienna. In 2019, Lydia was elected to the American Institute for Medical and Biological Engineering, and in 2020, she received the American Chemical Society's Biotechnology Division Young Investigator Award. The main goals of Lydia's labs are to improve the understanding of fundamental biological mechanisms that have biotechnology and biomedical applications. And today, she will be highlighting the importance of transcriptional regulation, adaptation in response to stress, as well as provide new methods for characterizing and predicting RNA network interactions and topologies. Lydia will also speak about the challenges of achieving equity in science exposure and how she engages families and communities to participate in her Raising Future Scientists program. Thank you, Lydia, for joining us this morning. Over to you.
Thank you so much, David. That was very kind, and good morning to everyone. So I'm going to share my screen here. So, sorry, it started in the wrong slide. So I was already said, my lab works on understanding basic aspect of cellular regulatory networks with the hope that one day, as this engineer here in the screen, we can actually engineer and optimize all aspects of cell biology. So this includes gene expression, this includes at the level of translation with the ribosome, with its speed, with the chemicals that it can process at the mRNA level, and at the level of protein synthesis, et cetera. Of course, these are highly regulated processes that we don't fully understand yet. And so moving the needle towards understanding much more of the fundamentals that underline these processes are key to be able to really truly engineer and envision cells in a whole new way. So today what I'll do is I'll do a little bit of an example of some of the work we've done in the context of an organism that has received quite a lot of attention in the field of biofuels. So although biofuels is not our main thrust of work, I illustrate this example to give you a couple of the major challenges and opportunities that lie in this field. So if we think about the bioeconomy, microorganisms, the view and the dream of them as factories to make any chemicals and any biofuels, any fuels that are currently made from oil, it's really key in our pursuit of trying to understand how to reprogram these organisms. This has become even more exciting as we have been able to expand beyond model microorganisms that have been discovered either for a while or recently discovered based on all the technologies that we have at our disposal now. And today what I'm going to be talking about is one specific one, Simomonas mobilis. So it turns out that Simomonas mobilis was isolated from alcoholic beverages quite a while ago. And as such, it has become central for a while in our efforts to produce biofuels. But when we think about the challenges when you're combining living organisms, living systems, and producing chemicals, or call it biofuels, call it any chemical, one of the major barriers in here has been toxicity. So this was, it's a very complex problem. If you can imagine when you are putting in either exogenous chemicals or making chemicals within these biological systems that are not built in such a way, there's several processes that are basic to the survival of these organisms that get disrupted. So just a few to remind you: DNA breaks, membrane disruptions, changes to the pH, inhibition of key enzymes, etc. So Simomonas turns out has figured all of this out. So this was initially, again, a bug that was already discovered to be able to withstand very high levels of alcohol. It is one of the favorites to make tequila, among other things. But it really, in our case, when we started this work, thankful to our career word back in 2012 or so, we were really inspired by this organism, but really thinking about it as a special system to start figuring out complex patterns of regulation. So we thought this was a great opportunity. It was a non-model organism to not just understand perhaps new fundamental biology, but also to develop sort of more creative strategies and address some of the challenges in characterizing regulatory bacterial networks. So just a little bit about Simomonas. One of the things that I don't want you to take for granted is that it's actually tractable. By the time we started working on it, its genome was known. Turns out a smaller genome than, let's say, a model like E. coli. And this actually was challenge number one, was actually done for us. A big challenge here in microbiology and understanding these systems is that most of the bacteria that we really want to understand that are doing the unique chemistries are not really tractable systems. So this is actually a huge area for opportunity in this field, but Simomonas was when we started working on it. We knew its genome, we knew we could grow it. Again, another challenge that we don't take for granted. And most importantly, it has this very unique enter-Durov pathway in which it is very efficient at taking sugar, metabolizing it to pyruvate, and making high levels of ethanol. So this is special when it comes to many standard organisms, including yeast. And even more special is the fact that when you're trying to break things like corn and produce byproducts like acetate and furfural, it also has a membrane that is well suited for these type of stressors. It's commercially easy. It grows under non-special conditions, pretty average temperatures, not very prone to phage contamination, unlike other sort of biofuel favorite bugs. And more recently, the value of this bacteria has extended from ethanol to other long chain hydrocarbons like farnesene. So again, this is one of the challenges even in picking an organism that is not a model that we hope we can continue to address in the field as we need to really continue to expand to other unexplored organisms. So the question for us that became pretty fascinated was, how does Zymomonas regulate its global metabolism to enhance ethanol tolerance? We knew this was a complex mechanism. We knew there were pathways involved. So this is where the story started. So this was also at the time where we were more and more realizing that RNAs are not just simple messengers here to make proteins. Indeed, more and more emphasis on RNAs as decoding systems that highly regulate many enzymes, many pathways, particularly under stimuli and environmental stress, was coming in the literature. So we also knew that this was a very complex world, which meant lots of opportunities for innovation and discovery, especially when you even throw the extra layer that these RNAs, we know, have a very extensive vocabulary of sort of bases and chemistries that they can incorporate that are also influenced by external stresses. So we were really interested about this aspect of decoding biology, right? A lot of the emphasis so far has been done on encoding, building DNA, building synthetic DNA, putting it into cell, transforming it, but we were really interested on this piece of decoding. I should mention that lots of bacteria, these are just some of the favorite ones in the world of biofuels. R stands for Rnomics, so this transcriptome discoveries of all kinds of RNAs were already there, as well as proteins. So the field has also been picking up quite a lot in terms of the discovery. The challenge here has always been, since the existence of new high throughput technologies, to look at RNAs and proteins, which ones are functional and how are they functional. So to lead a little bit into RNAs and what they do, I'm going to specifically talk a little bit about these regulatory bacterial small RNAs. So this is the mechanism, some of the basic ones that are known. Here they're represented in blue, and essentially what they're known to do the most is target mRNAs. And when they target mRNAs, whether it's at the 5' end in the middle of the sequence or at the 3' end, what they do is they hybridize and they change the properties of that mRNA so that either ribosomes are blocked, so you prevent the downstream enzyme of protein from being produced, or you change the half-life in the cell either directly by binding and stabilizing that mRNA, which means that mRNA is now longer in the cell to make more proteins, or by binding and leading to degradation directly by recruiting enzymes. So it's a huge effect on these mRNAs. And what is really, really exciting about these mechanisms is that these sRNAs, which could be up to 400 nucleotides or so in bacteria, can bind multiple mRNAs at a time to coordinate multiple metabolic pathways at a time. So there are several of them that we knew existed in E. coli that are well characterized, that participate in regulating oxidation and nutrient deprivation and iron deficiency. So it really wasn't unreasonable to expect that some of these regulatory circuitries will also exist in an organism like Zymomonas. So this is where we kind of led to, can we discover any of these in Zymomonas? And we started this work by really producing the native conditions that we would produce differential levels of ethanol. So aerobic conditions are known to produce a little bit less ethanol and really high levels of ethanol, 10 grams per liter or so when you take oxygen away. So our thought was, well, if we repeat the classic experiment where we just let the cells tell us what they're doing, maybe we can put them under high and lower ethanol conditions, sequence them, get their RNA, and start really understanding what types of RNAs, what types of regulatory molecules are being natively produced. So I should start by pausing and saying that this is what I just described sounds really easy until you do the experiment. And then you get 400 candidates and you have no idea what to do with them. So this is one of the big challenges here when it comes to a lot of these omics experiments, is this whole problem of having a lot of data and not having enough tools, big data, algorithms, pipelines to really interpret the data. So in our case, even working in bacteria, we had about 100 candidates here. 50% of those, after laborious experiments, we couldn't even detect. There were artifacts of the data, perhaps degraded products, perhaps truncations. And out of the ones we did pick, none of them had really any effect on ethanol or the phenotype that we really wanted. So this is at the moment where you're not the most popular advisor and you have to start thinking of other things to make your students happy. So this challenge of sort of finding the needle in a haystack, as sometimes I like to describe it, led to really good opportunities in the lab to start thinking about bioinformatics, machine learning, and really starting to apply it to this problem of how do we parse out all this big data and start really finding molecules that correlate to a phenotype that we want. It turns out it's a huge problem that traverses through a lot of these efforts in characterizing networks and regulation. So we needed systemic tools, and we really wanted things that we can generalize across bacteria. So I'm just very briefly going to describe to you one of the first opportunities here that we got to really think about how do we use functional genomics and how do we use sort of this idea of conservation in the genome and look at all data that was already there for all many species that had already looked at for pathogenesis purposes in the medical community, and how do we really incorporate that into sort of machine informing algorithms to do the best predictions we could for RNAs that actually affected what we wanted, in this case, ethanol. So there were several hypotheses here that had to do with expressing these RNAs, really mapping regions that were really likely to give us real candidates, and then using prediction tools and treating them as a network and looking at those functional correlations so that we can get the best chance at identifying true regulators. So we call this one of the first pipelines that we designed refine. And this was actually excellent in the way that we could go from hundreds of candidates to tens of candidates that could actually be biochemically explored. This was very successful when we applied it to Cymomonas. We have since applied it to other organisms. And one of the success stories I'll tell you is the discovery of this one RNA here, this CMS2, 6, and 4. So if you especially look at these 6 and 4, one of the things that you see right away is that these were some that we confirmed. This is a northern blot. So the band indicates its real presence in the cell. And as you can see, the thickness of the band really indicates that these are hugely expressed in the presence of ethanol. So they seem to be ethanol dependent, not so much the oxygen that we use to create these conditions. So we became really excited that we at least were overcoming partly of the challenge of what's real. So they seem to be real and again, naturally upregulated in conditions of ethanol. The next thing that we did was kind of go back to traditional genomic approaches and clone them and overexpress them and delete them. And one of the things that we found that were also very encouraging is that if they were deleted from the cells, as you can see, these yellow bars here, it's growth under ethanol versus the gray bars here is grow under ethanol. These RNAs seem to be really important for the cell to be able to grow under ethanol. Overexpressing them, not so much, but deleting them definitely had an effect. So we were pretty encouraged that this could be real. And then even in ethanol accumulation. So when we would measure the type of ethanol that was naturally expressed, these deletions here, these delta 4, delta 6 especially, had much more decreased levels than when the RNAs were in the cell. So this was a good sign. And then became the second challenge. Well, what are they doing? And what are the regulatory networks? So this is another big challenge in the world of characterizing these regulators, is that they very rarely work alone. What we know is that they're very part of intricate, complex networks. And to start deciphering these pieces, especially in their relevance to in vivo cell biology, is quite a challenge. So this is where a couple of very specific challenges that relate to the molecular description of these in vivo is which targets are direct as versus which targets just get secondary sort of effects in the cell. That's a hard question to answer. Typically, they have many challenges, and these stress responses are very complex. Then it's sort of the mechanisms, right? There is especially quite a large and diverse types of mechanisms here, and we don't even know all of them. And lastly is, how do they bind? What interfaces do they bind? And these are definitely some problems where machine learning has also helped quite a lot of our data interpretation. So we did a classic. We actually kind of came up with what we call a 4-omics approach, which we combine mutations, we combine time dynamics, we combine proteomics, we combine RNA-seq, mRNA-seq, and pull-down experiments to physically grab RNAs and assay what they were binding to. And this was another method that turned out to be quite effective in letting us know at least the seeds of these networks to start studying. So along the way, in answering the question of, well, what are the direct targets and how do they operate? We also have been able to incorporate a little bit of synthetic biology here, again, to address this challenge of how do these RNAs function in vivo. So one of the hypotheses that we've come out through our work in recent years is this idea of measuring more of the structural accessible configurations of these RNAs. So we've been really trying to incorporate information at the molecular level, thinking that these RNAs are highly dynamic, and that if we can actually figure out which sites are either really available for binding or really occluded, we might be able to start getting to specific submolecular structures that are important for function. So this is another opportunity where we brought in a technology that we've named Interface. There's several iterations of that we've done in the lab, where we have combined synthetic biology, the ability to create and monitor these loops of RNA, synthetic RNAs in cells, and use them as a reporter to tell us exactly those available regions for functionality. So combined, this was great. We can produce now maps like this, where we can get now information from omics, and we can get in functional molecular information, and we can start creating maps and start really betting, making high bets on which of these regions are regions to track for functionality, which has been really helpful, because what we've been able to now do is use predictions, filter them based on a lot of these functional screens that we've created in vivo, and come up with likely pairs. So at least likely pairs of RNAs and their target mRNAs. So to make here a conclusion of the story, this has been really helpful. We have been able to map targets. These here are EMSA, so these are binding assays. And when you see this higher band here, this is radiolabeled. That means they're forming a complex with these particular mRNAs. Two of the mRNA targets that I'll tell you today are these alcohol dehydrogenase and aldehyde dehydrogenase that we have to be target. We have confirmed that they work in vivo by actually labeling, taking the regulatory regions that we've now mapped and putting it under to control GFP expression. And we can see decrease and increase of these reporter in the absence and the presence of the RNA for these genes. So essentially what we have determined is that if you can see here, this is the wild type, we knock out this RNA regulators, you can see a big impact on the expression of these targets when these RNAs are gone, meaning that they're having an effect in vivo regulating them, which was pretty exciting to map out. And so what turns out is that two of these targets are very, very interesting targets. These two targets that I told you, they're part of bigger networks of targets that now we have figured out. And the first things that we see is that they have completely different roles on this target. One stabilizes, the same RNA stabilizes this target, but degrades this target. And these target, it's a huge node here in sort of a little valve that controls how much ethanol is produced. So as ethanol is being made, some of the targets, at least this two, what it's doing, it's regulating that balance, almost telling the cell, okay, you have the go ahead to make more ethanol, or this is already enough, back up and send it back to acetaldehyde. So this is one of the valves that has been highly regulated by RNAs that we have uncovered. These are some of the structures of these RNAs. So you can see they're complex. They're like little octopus, multiple binding sites. They bind multiple targets. And one of the most interesting thing about this system is that this led us to uncover the first dynamic duo of RNAs that really work together. This is not something we've seen in other systems before, but you can, if you pay close attention here, what you can see is that the ZMS4 here has a very distinct binding site for this RNA. And we're still trying to really figure out what that dynamic means, but it's really fascinating that they have such a broad range of target and bind each other. And lastly, when you put it all together, we just talked about these right here in terms of these alcohol and ethanol metabolism, but in reality, lots of pathways here that they have target and that they regulate in vivo have to do, again, not shockingly, to many of these phenotypes, many of these pathways that are involved in granting cell ethanol tolerance. So lastly, we have been able to say, well, if we can do that, if we can characterize them, then let's imagine these as strain engineering tools. Let's imagine that we can now manipulate them. We can increase the numbers and we can rationally design these RNAs to produce cells that do what we want them to do. And what we've done, it's some good news, is make combinatorial effects of these RNAs, and we've gotten up to 10-fold improvement now, pushing the limit of how much even ethanol these cells can take. So they grow better when we make these combinatorial approaches, now that we know what these networks can do. So not new, RNAs are being used highly and widely in technologies and in making strains. And lastly, this is it's also led to some biophysical model where we've really started to define much more carefully the thermodynamics of binding between RNA and RNase in vivo, using these ideas of accessible regions that are dynamics that change with time, etc. So Racing Future Science is the last thing I'll tell you about. So this is our hypothesis has been for a while that family support is key in underrepresented students for them to come to STEM and stay in STEM. So we started this experiment, to be honest, mainly because my mom retired and I needed to find her something to do. And she was spending quite a lot of time in Austin. So I figured I'd use her Spanish skills and connect her to some other parents to talk about what it was to raise a child that wanted to do something completely different. And that she loved it. It turned out from that I said, well, let's actually create this into something formal. And so far, we've been able to really work steadily with about 250 families where we treat them like a cohort. So we follow up these students when they're in middle school and we bring them to the lab and they come in and they really talk about what it is to be a scientist. So Jennifer, your talk reminded me of something that we do, which is that they do quite a lot of artwork and painting themselves in science. And they wear their lab coats and their goggles and they start really with that visualization parts. We do interviews with families to customize a little bit what are the barriers for that specific family and target that a little. And then they come and we really have a blast talking about the world challenges and how sciences can help, etc. So with that, I thank you so much for your time. In summary, these environmental changes, metabolic stresses are exciting in terms of biology and fundamental biology. We've kind of taken a road where we've been forced to think creatively about innovating new ways to study them and create new technologies to understand a lot of these effects. And of course, this has been a wonderful opportunity to use this NSF funding to also inspire a lot of the younger students in our community. So thank you so much for your time. This is our group, and I'm happy to answer any questions if we have a little bit of time left.
Great, thank you, Lydia, for a wonderful talk. So we will be taking questions from the audience now. As a reminder, you can do that through the Q&A box at the bottom. So the first question is, you gave some nice examples of how interactions among individual molecules form the basis for complex interaction networks. How feasible would it be to re-engineer the configuration of a network by re-engineering interactions?
Yeah, so we think it's feasible. I mean, in the example I gave, we just played with expression levels and tuning these expression levels of these molecules. But in other systems that we have where we use more of carbon starvation in bacteria, that's a lot more tractable. We've been able to do a lot more creative things, like actually take those RNAs and completely redesign them to create a gradient of activity. So we have those RNAs that we can make more active, less active. We can preferentially target them to recruit certain mRNAs, et cetera. So we think it's definitely feasible. The bottleneck there, it's how well can we manipulate the genetics of that organism.
Great, thank you. And with that, I'm going to pass it off to Stephen Zender, who will be introducing our next speaker.
Thank you, Adrian. It's my pleasure today to introduce Dr. Julius Lux. Julius is currently an associate professor and associate chair of chemical and biological engineering at Northwestern University. He received his PhD in chemical physics from Harvard in 2007 and went on to pursue a postdoctoral fellowship at UC Berkeley, where he helped to engineer versatile RNA sensing transcriptional regulators. Along with Jennifer Doudna, Leo Pachter, and Adam Arkin, he led a team that developed ShapeSeq, which is an experimental technique that utilizes next generation sequencing for probing secondary and tertiary structures of hundreds of RNAs in a single experiment. Today, Julius will be addressing how cells sense and respond to their changing environment, as well as describing how RNAs facilitate these roles in the cell. By understanding these fundamental questions, synthetic biology techniques can be used for low-cost, rapid, and deployable diagnostic platforms to tackle emergent challenges such as those that we've seen in the current COVID-19 crisis. Julius will also be speaking about new entrepreneurship avenues to deliver these technologies from the lab to the world, contributing to new opportunities in synthetic biology graduate education. We are absolutely delighted to have Julius here with us today. So thank you, and feel free to take it away.
Thank you so much, Steve. As I get set up, I have to say this is an incredible honor to be invited to do this and to share the stage, this virtual stage, with these scientific synthetic biology rock stars. I feel very lucky. Also, it's really a pleasure for me to be here in front of NSF. I mean, NSF has been an essential supporter of all of our work that's really spanned fundamental biology investigation, synthetic biology application, technology commercialization, and as Steve mentioned, some new approaches to education. Without that, none of this would have been possible. So as Steve mentioned, the story I'm going to tell you today really starts off with a basic biology question. How do cells sense and respond to their changing environments? And it's a story that's going to take us from discovery all the way to design. And I'm going to tell this story in the context of something very, we like to think about in terms of synthetic biology, this design, build, test, learn cycle. But I'm going to add a couple of elements on applying and teaching. And throughout, really acknowledge how NSF, the investment of NSF has been central to all of these developments. This, beginning with learn, these fundamental questions, biology does amazing things, and synthetic biologists are really here to try to understand these features of cells, to understand how we might repurpose and re-engineer them for our own uses. And of the many things cells do, much like Jennifer, this ability of cells to sense and respond to changing environments is just absolutely fascinating. And what I like so many things about Jennifer's talk, but in particular, her breakdown of how to view some of these phenomenon is very similar to how we view this. You have a very complex behavior, but we like to think about it as occurring under multiple spatiotemporal scales. A molecular level binding event is the first thing that really needs to trigger to happen. Maybe a small molecule or chemical binds to an RNA or protein and changes that molecular structure. This leads to a gene expression change, much like Lydia mentioned already, that eventually feeds into a genetic network, which is a different scale of biomolecular interactions that enable cells to process that incoming information and make decisions about how to change their physiology or motility and these other behaviors that happen on the cellular or even biological community scales. And so as we've tried to understand this basic process, this framework has really helped us. And much like Lydia, where we find ourselves most fascinated is at that molecular scale, and in particular trying to ask, how can RNAs play a role in this fundamental sensing and information processing capability of cells? Well, there's many RNAs around, as Lydia just said. One particular class that we are fond of are called riboswitches. These are really special and ancient molecules that can sense and bind to chemical ligands, but then dynamically alter their structure in response to that binding event. And that structural change change acts as a switch, usually for gene expression processes. And as you know, biologists and synthetic biologists, we want to understand how these things work. And it eventually leads us into very basic questions about RNA folding and how RNA folding occurs in the very complex cellular environment. Now, one of the things about cellular RNA folding that to me has been one of these mysteries that we haven't known much about is just how dynamic it is. In fact, RNA folding begins during RNA synthesis. And to kind of illustrate this, I love this artist's conception of the transcription process to help you get your mind into the wonder of this. So this is a picture of RNA polymerase in blue, bombing down a DNA track in red, and then shooting out of the back is this yellow RNA molecule. And what I love about this video is it captures the dynamics, how fast it is. So you got about 10 milliseconds per nucleotide and microseconds for some of the basic RNA folding events. Now, what this doesn't capture, though, is some of the other complexities. Transcription is not smooth. It's often a jittery process. You can have other RNAs and proteins binding to the RNA coming out during transcription. And the RNA molecule itself can actually dynamically rearrange during this transcriptional process. As a result, we know a lot about the structural biology of the polymerase and DNA, but almost nothing about the RNA molecule. In fact, the artists kind of put this spaghetti thing in the back because they didn't really know what else to put there, but that's likely not the case. So to investigate that, we've really been trying to turn the problem on its head, and rather than thinking about following the folding trajectory of a single molecule in time, rather fire up a population of RNA somewhere in the middle of active transcription and taking a structural snapshot of all of them, and then reordering those structural snapshots in the order that they must have occurred in to reconstruct this RNA folding pathway. Well, as Steve mentioned, you need a couple pieces of technology to do this. One is, how do you take a structural snapshot of hundreds of thousands of RNAs at once? And this is where this shape-seq method comes in. It utilizes a set of chemistries that covalently modify RNAs at unpaired positions. You modify the RNAs, you then use next-gen sequencing to map where those modifications occurred. And since you know something about the chemistry that you get more modification frequency at unpaired positions, you can infer the structures of these molecules from this data. We then couple this to transcription reactions, and we get this data for every single length, intermediate length of the RNA during transcription. You get these matrices, which is kind of shown on the right. The length of the rows in the matrices grow in length because the RNA is growing in length, and then you see these changing patterns and reactivities that reflect how the RNA molecule may adjust itself during transcription. Now, it's beautiful data. It's a lot of it, and it's really hard to understand by eye what's going on. And so with support of the physics of living systems, we've been developing algorithms that can take this data, merge it with our current understanding of RNA folding to come up with experimentally informed two-dimensional and three-dimensional pathway predictions of these folding processes. And to give you a sense of what that looks like, this is the application of that technique to a very ancient RNA molecule called the signal recognition particle RNA. That's an essential molecule in all living cells. What's fascinating about this molecule from a folding perspective is it actually ties itself into a knot during transcription, and it has to figure out how to untie it. And what we found is a very cool molecular mechanism by which this molecule can internally bind itself and overcome what looks to be an insurmountable structural barrier to finally zipping up into a long-range helix. This type of phenomenon is actually called in the field, the little jargon here, toehold strand displacement, but it's a technology that's actually been developed quite extensively synthetically. But this is the first observation that it occurs in one of the most ancient RNA molecules that we know about. So this is kind of a cool feature of natural biology we're learning about. And it's an example of one of these design principles where we are understanding, the rules of life in NSF parlance that we can then use in a synthetic context. Now the question, and I should say, going back to the riboswitch story, we can now do the same experiment with this fluoride anion and ask, how does the binding of this ligand change the structure and implement the genetic switch? And in fact, we get high enough resolution that we can identify the nucleotide that, when it's transcribed, is the decision-making point of this molecule. So now that we have a deep understanding of this mechanism, we've learned a lot. Can we use this to design, and looking towards using this knowledge to benefit society? And the way I like to, view this is like, we're translating our basic biology question, how do cells sense their environment and make decisions with that information, into a synthetic biology question. How can we use cells to sense our own environments and make decisions with that information? And this is really the ethos of synthetic biology. We want to learn how we can repurpose these same molecular machines to enable us to monitor these chemicals in our environment. Well, this fluoride riboswitch, I'm telling you this story because it's the natural bridge here. It turns out that fluoride is a global contaminant of concern in water in certain parts of the world. In the US, we tend to put it in our municipal water supplies at low levels to harden our teeth enamel. But at higher levels, fluoride actually gets into your bones and causes a debilitating condition called skeletal fluorosis, which is lifelong. And it's a big deal in many parts of the globe. This kind of got us into thinking about water in general, and the realization I think we all know is water is absolutely essential to almost everything we do in our lives, from cooking to cleaning to producing our food, transportation, you name it. We use water to do that, and at least in the U.S., many of us I think take it for granted that our water supply is clean. Things like Flint, Michigan, and Newark, New Jersey obviously started to shake some of that confidence, but by and large, people assume everything is fine. But that's actually not the case. In fact, at a global scale, it's estimated that over 2 billion people lack access to sufficient amounts of clean water for daily use. And it's actually one of the United Nations Sustainable Development Goals to try to fix this problem by 2030. Now, part of the challenge with water is that as humans, we can't often see or taste or smell contaminants that even at low levels are harmful to us. And if you look at the contaminants, these three are the top three from a global health perspective. But under the hood, there are many, many others. This is a list of regulated chemical contaminants from the US EPA. And what's not even shown on this list is hundreds to thousands of others unregulated chemical contaminants that are so-called contaminants of concern. As you can see, you don't have to be necessarily a chemist to appreciate this is a really complicated problem. It's a vast space of chemistries that you need to be able to detect. But it motivates us to ask this basic question: shouldn't we all know what is in our water so we can either change our behaviors or demand more clean supplies? Now, it's not like you can't measure water quality. In fact, these are wonderfully sophisticated instruments to do just that. They're specific and accurate. The problem is they're instruments. They're expensive. They're expensive to operate. You have to have expertise to use them. And as a result, they kind of sit in a laboratory setting. And it's really a centralized testing infrastructure. And that infrastructure just can't meet the scale at the global level that we need water quality monitoring. So, we like to ask, how can the synthetic biology community help? Or how can we make water quality monitoring accessible for all? Because it's oftentimes the underserved portions of our population that are suffering the most from these contamination issues, but they don't even know about it. Now, I've said this already, but this is what we try to do at synthetic biology. We've studied these molecular sensing mechanisms. Can we repurpose them so that we can use these same machines to sense our environment? Can we rewire them so they produce nice visual outputs rather than the genes that they naturally control? And can we re-engineer them so that they don't function within cells, but instead in these cell-free systems that are much easier to use? In fact, they're so easy. Once you figure out how to assemble these reactions, you can freeze dry them just like you make astronaut ice cream, and then put a drop of water in when you need them at the point of use to make this happen. So we've been exploring this recently. This work has really been enabled by the career award that I've been lucky enough to have. In the context of this fluoride riboswitch, here we rewired the riboswitch to control a protein that produces a nice visual detectable signal that's a yellow color. And when we configure it in a cell-free gene expression reaction, it turns out this thing senses fluoride down to about 2 parts per million, which is the EPA secondary toxicity limit. So this is potentially a very viable method for sensing fluoride. Going beyond RNA mechanisms, there's many, many proteins that actually sense these small molecules, as Jennifer started to point out. A particular class we like are called allosteric transcription factors that we figured out how to utilize. And here we're moving away from protein outputs and getting into fluorescent RNAs. And so by eliminating the translation step, we can speed up the response. And in honor of Rosalind Franklin, whose work has basically enabled this, we call this the Rosalind platform. What's cool about Rosalind is if you want to sense a different compound, you just change the protein and rewire the DNA. And so we've been able to sense things from antibiotics to personal care compounds, cleaning products. And on the bottom row, what's most people in the US are interested in are metals, such as lead and copper. And these are pretty accessible. This is just a little video showing the green color that's produced. We built a little 3D printed handheld illuminator, uses some blue LEDs and stage light filtering. And on the tens of minutes, you're getting a nice yellow color. We just put out a preprint yesterday showing we're down to about 5 minutes for this color change. And what's cool is that, people can get this. In fact, Scientific American picked this up in December, and the Discovery Piles picked this up, also calling it taste tubes, which I thought was a pretty interesting way to communicate to the public the power of synthetic biology, potentially for solving a problem of theirs. But we need to go further. This is all in the lab. We need to at least try it out into the field. And while we were doing this work, actually, there was a major catastrophe on the West Coast in the town of Paradise, California, which is obliterated by a wildfire. And with generous support from NCB, we were able to get a rapid grant to try out this technology in the context of Paradise. The problem was that the fire was so hot, it basically melted all metal in that town surroundings, metal pipes, car batteries, you name it, and tons of metals in the environment. We wanted to see if something like Rosalind could help with remediation efforts to identify sources of contaminants. Now, before we went out into the field, we needed to do some basic testing. Is it really easy to use? And so this is the test I like to call the 5-year-old test. Is it simple enough for a 5-year-old to use? And it's called the 5-year-old test because that's the age of my daughter. And in fact, she can put a drop of water in a tube and it works. So that's awesome. We then took it out into the field and put it to a more stringent test we like to call the PI test. If it's really easy to use, even the PI can use it. And that's me, I did it. And some of the dramatic pictures we observed in Paradise. This was really a good thing because we actually discovered a major failure mode in the reactions that we were able to fix back in the lab and ultimately show that these tests function as designed in municipal water from Paradise to identify high levels of copper and zinc. Now, it's one thing to show that it works, the experts can use it in the field, but we really want to get this out into the public. And so we were starting to think about how we might do that when something else happened, which is why we're all doing this virtually, which is the coronavirus pandemic. And one thing that was interesting, tragic in a way, but a commonality between global water testing and global coronavirus testing is the limitations of laboratory-based testing and kind of the traditional view on it, how it just wasn't scaling to meet the need. Now, I believe Doug is going to talk about a really cool approach to solve this challenge with some of his techniques. What we wanted to explore is whether or not we could try to use the same type of thing, easy to use point-of-care diagnostics to address the coronavirus diagnostic issues. And actually, we assembled a team across the Northwestern Center for Synthetic Biology, kind of start to finish. We had computationalists modeling different formulations of these reactions, suggesting which ones we should build in the lab, what optimization schemes we should do. We learned a ton about isothermal amplification techniques and CRISPR-based detection, and we're able to come up with a diagnostic that was down to the low femtomolar range, and now we're in the tens of attomolar. At the same time, we were thinking, there's only so much an academic lab can do. And so we wanted to create a commercial venture that could basically deliver these tests to those that needed most. And so this STEM loop was created out of the Northwestern Center for Synthetic Biology, basically to take this biosensing platform and get it out there for community health. And there's been generous support from SBIR programs to help us launch this in the middle of the coronavirus pandemic. Now, this has been a really cool story, a scientific story, but how can we use this, what we've learned, to generalize this to be able to teach the next generation that's going to fuel the bioeconomy? And here again, I have to point to the Career Award for really sowing the seeds of this. One of the educational aims of my Career Award was to help found and really develop the Cold Spring Harbor Synthetic Biology Summer Course. And this has been such an amazing adventure. And as the years have gone by, to see the people that have come out of the course has been super special. One I'd like to highlight way in the back is actually Khalid Alam, who's the co-developer of the Rosalind platform and now the co-founder and CEO of STEM Loop. Also in the career award was the first image that I actually showed you. This idea of breaking down biological systems along scales. This was actually a class I was trying to develop to try to be able to map how can we use core engineering principles, things like thermodynamics, kinetics, and transport, and map them onto biological systems so that we can utilize the insights gained at the appropriate scales and then connect them, connect the dots. This has been revolutionary for our own research, but we thought this would be a great way to think about reframing graduate education in synthetic biology. And in fact, it's these concepts that we were lucky enough to kind of crystallize into a training program that we were lucky enough to get a new NSF NRT program based on this concept. And the idea is that grad students should typically come in their home departments. They kind of specialize early on, but they kind of get connected at a research level, but it's a missed opportunity to give a real convergent training program. And so we actually have them come in and first learn this deconstruction approach. Through case studies, they break down different synthetic biology technologies along the scales of phenomenon that are needed to put them together. And this importantly also gives us an important hook to weave in things like ethics and socially responsible research throughout this. And so we ask deep questions about that. Students can then, specialize in courses and scales that are going to be important for their own graduate thesis work. But this framework of deconstruction really, we think, is going to help them facilitate collaborations throughout. And this has been an absolute delight. I just finished this deconstruction course last week, and what was surprising to me was how natural students really caught on to this approach, and many were empowered. So I've been lucky enough to tell you about a vignette that goes, from design all the way from discovery all the way to design. And again, this would not have been possible without the amazing collaborators and trainees that I've been lucky enough to be a part of, and also NSF. And I listed a few NSF people near and dear to my heart there. Teresa Good has been with me from the very beginning, and David Rockcliffe, Steve Peretti, Kristen Blagoev, and John Weishamble have been amazing program officers on some of these grants that I've told you about. And with that, I'd be more than happy to take questions.
Great, thank you, Julius. The first question is, you talked a little bit about democratizing synthetic biology. What are other opportunities for incorporating citizen science in the field of synthetic biology?
That's a really great question. I think, there's lots we're really interested in these kind of cell-free synthetic biology approaches, from education to allowing people to really touch biology with their own hands and explore these concepts. I also think, and that's cool, there's lots of ways to get involved, and we have ideas of citizen science. We're spinning up ideas in our minds, for example, to get these water quality monitoring devices in the people across Chicago who are really interested in Chicago river quality, and get them to ask questions. To go back to something Jennifer said, though, I think it's really important to go beyond just, can I do a cool demo of my particular science for people, and really start to think about how to have more conversations and more listening opportunities to help people engage with what is even possible with synthetic biology. We had a really amazing guest lecture in our course as well from Laurie Zoloth, who's kind of leading bioethics person in the synthetic biology space. And one of her real take-home points in my mind was this concept of, why don't we listen to the general public and understand what their needs are, kind of like what Jennifer said, and think about how we may pivot our technologies to address those needs. And I think once we have that kind of two-way conversation more often, I'm hoping that we'll find more buy-in and excitement for the field.
Great, thank you. With that, I'm going to hand this off to Mitra Basu, who will introduce our final speaker for the day.
Yes, so it gives me great pleasure to introduce Doug Densmore. Dr. Doug Densmore is currently Kern Faculty Fellow and Associate Professor of Electrical and Computer Engineering at Boston University. After receiving his PhD from University of California, Berkeley in 2007, he became a UC Chancellor's postdoctoral researcher at Berkeley, where he studied the development of system-level design methodologies for electronic systems. Doug is also the director of the cross-disciplinary integration of design automation research group at BU, where his staff scientists, postdoctoral researchers, graduate students develop computational and experimental tools for synthetic biology. Doug has co-founded 3 commercial synthetic biology-based companies, and is the co-founder and board member of nonprofit NONA Research Foundation and Biodesign Automation Consortium. Today, he will be presenting on how NSF supported efforts have helped to develop a wide variety of biodesign automation software, principles, and workflows, including the design of biological genetic circuits and hybrid biological electronic microfluidic systems. In addition to his scientific research, Doug will present the STEM pathway program and international workshop on biodesign automation, which are dedicated to fostering the next generation of synthetic biologies. It would have been very nice if all the speakers were here at NSF. That would have been in person to see all of them and talk to them. But anyway, we do whatever we can. So with that, I hand it over to Doug, and thank you, Doug, for making time to come here.
Well, thank you, Mitra, for the introduction. Let me get my screen pulled up. Okay, people should be able to see that now, so I'll get started. So this is great. Like Julia said, it's really fantastic to be here. I still think it's a little strange that as an electrical engineering PhD, I come and talk to people about how to think about designing biology. But this talk is meant to get at that a little bit, and it's meant to get at, if you think about designing systems that we created, that being electronics, how could you think about that with systems that we didn't inherently create, that being biology? And so I'm going to talk about some of my journey through that with NSF and my particular viewpoint on how we engineer biological systems. So really quickly, I just want to mention, as was pointed out, there's some things I have potential conflicts of interest with, both commercially as well in the nonprofit space. But I think it's interesting to note as well that NSF has been very helpful, fundamental really, in me developing some of these. So I'll talk about an SBIR-based approach to funding a company called Lattice Automation. The National Science Foundation Advances in Biological Informatics supported the NONA Research Foundation, and also I get numerous grants every year for workshops, and some of them go to the Biodesign Automation Consortium that funds the IWBDA workshop, which I'll talk about. So again, to my background, what I come from is probably the least sexy electrical engineering field, and that's electronic design automation. That's the software that builds the computers that we all use. And it was built in a bottom-up way, where we had individual semiconductors, and we slowly exported up the physical design process, the logical design process, then kind of the specification process. And we use it top down. And you can see in that process, there's a clear specification portion, a design portion, and a build, and then testing portion. And that's a very simplified design. If I were to go to an EDA conference and show that, everyone would kind of dismiss this because it's extremely outdated. If you look at how electronics are designed now, it's much more complicated, where there's not a clear delineation between what is software and what is hardware and what is off the shelf versus what is custom. And it's become a very complicated ecosystem that really stands on the shoulders of giants. But what's particularly interesting to me is that there's a clear point in this design path where what is done in hardware and what is done in software is kind of translated from a high level specification. And that kind of motivates a lot of my research. And so what I'd like to do is that my research would like to have functional descriptions of what we'd like a system to do. So a biological system might look like a circuit where we have some external inputs, and based on those inputs, we produce an output. We'd also like to be able to capture the structure, that if we put pieces of DNA together in certain ways, the circuit or function will actually be executed. But if we put them in the wrong way, it won't. So I want to capture function. I want to capture structure. And I also want to capture performance. It's not enough to say that I'm going to express this protein eventually. We probably want to be able to say when we might express that. And we can use temporal logics to do that. And then naturally, we also need to have a library of biological elements that we're going to put together. And that's going to realize those three different orthogonal specifications of functionality, structure, and performance. And so this goes into a magic black box in a lot of my work. And then a logic-based genetic circuit comes out. And that's a lot of what I've focused on, is how to do this process. And so what you'd sit down and do, and sit down in a computer, you describe functionally what you want to do, and you get a circuit out. And that looks very much like how we might do things instead of in electronics, but instead of getting semiconductors out, we get DNA. What I've thought about in the past couple of years, and what I want to share with you guys, is this vision that since we're doing this already at a computer, we're already making what I would call the wetware. We're already making the biological devices that are going to carry out this functionality. Couldn't we also create hardware at the same time? And that hardware might be a microfluidic device that's going to house some of this, the software that controls it, and then ultimately integrate all of these together in sort of a hybrid system that combines both biology, hardware, and software. And so a lot of what the flows at my group and what I think about is that we could start with high-level specifications of biology, talking about structural and functional and constraints, what we might want a system to do. Then it would go down a path that I've described and we'll describe in this talk that can turn things into wetware, makes the actual DNA that carries out that. But then make the microfluidic environments that are going to test and kind of maybe deploy these in a real environment. And then also make the control infrastructure. So not only do you get the microfluidic out and the DNA, but you also get the software that's going to control that and carry out your experiment. And that would be hardware, software, wetware, co-design. And so I've been fortunate to get a number of NSF grants that kind of helped me tell this story, starting in 2012 and then most recently with a SemiSymbio award. And so I'm going to speak to these during this talk. And the way that you would tell this story is not kind of chronologically in terms of how I got these grants, but really starting with maybe thinking about what's called the Living Computing Project. And that really lets you specify genetic circuits. Then Puppeteer, which comes from my career award, which is then once we have the specifications, how we automatically assemble them, then how we can archive and share designs, which I got through an NSF ABI award. And those would all kind of factor in to something like the SemiSymbio award, or an actual application of this to create a hybrid biological system that we could deploy into the environment. And then those kind of sit on this foundation, or what we can go out and do afterwards is disseminate this information through commercial endeavors, promote this through workshops, or educate through outreach programs. I'm going to talk about these efforts, but I'm not going to talk about the Clotho effort just because I only have so much time. So the first thing I'm going to talk about is the NSF Expeditions in Computing Award. The program manager on that was Mitra Basu, who was generous enough to introduce me. This is one of the larger size awards. It's an award that's really focused on how we can think about these three axes that I show in this figure. And that's how we can take paradigms like digital, analog, memory, communication, and then translate them to biology, where we can specify these designs, design them, and verify them. And then also importantly, how we measure them. This, if nothing else comes out of this work, what I'd like to have happen is that when people describe biological systems, specifically biological circuits, no one ever says this circuit works. You don't say that with electronics. You never say something works. That's kind of by default. You really say how fast this works, or what the throughput is, or what the latency is, or what the cost. You have a metric. And really what this work would like to do is quantify those metrics and try to apply them to biological systems. A lot of people involved in this grant, people at MIT, Lincoln Labs, BB and Raytheon Technologies, BU. And again, some of the examples there are how to take high-level functional descriptions that come from electronics and turn those into biology. Other things are these metrics. You know, again, the traditional way to think of a circuit is it carries out one function. But if you look at what other functions it might carry out, it actually might carry out other digital functions better than the one it was designed to. So we want to find ways to capture that. We also want to make these design environments, microfluidics and others, to carry out these, to make sure we have controlled environments so they can realize these functions. And then archiving all of this through registries and parts repositories that also quantify how these systems work. So the classic example that comes from this work that many of you might know me from is work that I did with Chris Voigt at MIT on a system called Cello. What's shown here is a graphical user interface. You sit down at a computer, you write code, in this case, something called Verilog, the same code that's used to make electronic digital circuits. You write that code, it's then quote unquote compiled to a library of DNA elements. And so you end up getting a transcriptional translational logic circuit out. But not only do you get one circuit out, it doesn't say this is the one design. It gives you a variety of designs based on a scoring metric. So it gives you not only all the designs that fit within the score, but also all the different structural orderings of these circuits based on constraints you also provide. So it gives you this design space, so to speak, of potential genetic circuits that will realize that high-level functional description. And we've written numerous papers on this, and you can find this code at the URL along with being able to use it. All of this stuff is open source. An extension to that is that as these circuits get larger and larger, one of the things this project thinks about is that if we have an extremely large, in terms of the number of gate circuit, effectively that may kill the cell. And so what we might want to think is decompose that circuit into multiple cells, and so that it's communicating intercellularly. So a computational problem that my group and others think about is, given a genetic logic circuit, what's the most efficient partitioning of that circuit in the submodules? And you might want to do that so that it has a variety of communication motifs between cells. And this was kind of the motivation for much of the microfluidic work that initially came to me, was that if we're going to have these cells communicating to one another, we're probably not going to spatially just put them on an agar plate. What we're going to want to do is actually have a controlled environment where the communication channels between them are physical, and we could get that same communication network out of a tool. So we got the circuit out of a tool, why not get the microfluidic out of the tool? And that's something that also comes about in this living computing project, is how to do this kind of partitioning work. We need to design these microfluidics. So some of the work, again, is how we make, what I do at the end of the day is I make CAD software often. And so we've made CAD software that lets you describe microfluidics. Here are two examples. Here are quick little videos. On the left is actually a CAD canvas where you can drag and drop microfluidic components and resize them and redesign them. So instead of using something like an Illustrator or a SolidWorks, which isn't particularly, it's useful for microfluidics, but it's not designed explicitly for it, you can do that work. And what's shown on the right is, then again, starting from a high level description of how liquid flows are related, merging, splitting, joining, choosing between them, selection. You can write another high level language similar to the one we made genetic circuits with and get a microfluidic out the other end. And the idea, again, for me, is that what I want to do is I want to make software that not only does this kind of in an automated way, but does this correctly by construction. So what it would mean is that you can't do things in a way that are going to end up producing devices that are going to fail either structurally or functionally. And we can do that by adding rules and constraints, syntax and semantics into these flows. So once we would have all of that DNA that we want to produce out of maybe a cello-like tool, it says, hey, here are 20 circuits you want to make. Work for my career award would help some of that. So my career award was one of the earlier things I got, and it was kind of prefaced on this idea that if I want to put this big piece of DNA together, I don't know if anyone watching this can just tell me how to do this. If you said I wanted to put all of these circuits together, and I asked you the most efficient way to do that, that could be potentially quite complicated. Not only complicated in terms of getting it right, but also in terms of how long this would take and also how much it would cost. So very early on, things that I was involved in was, if we think of these assemblies as graphs, and you think of the intermediates in the graph as being assemblies, it might behoove you to take longer to do a single assembly, because it might expose intermediates that you can reuse in other places. And since you can do that, you can then start to think about how you might automate the planning for these. And that planning became something called Puppeteer that was started in this career award. So this would take the designs that would come out of a design system like a cello, and this would be this graph. And you'd say, oh, this is simple. Just put A and B together to get AB. That's what's shown in the graph, this assembly. Very simple, putting two parts together. But in practice, the actual protocols that carry that out, if this is a very simple like enzymatic cloning operation, you're going to have to do a restriction digest. You're going to have to do a ligation. There are going to be some tests. So what looks like a very simple assembly becomes potentially a more complex protocol. And then once you add additional information on that about scheduling it, who's going to do it when this grad student doesn't want to work on the weekends, this person is busy, how is this going to work? This is what an actual schedule looks like. So what Puppeteer does is automate the planning of that. Now, the idea isn't to have grad students do this. These are figures from McDonald's. So what's shown on the left is how McDonald's could think about making their food. The rows are individual menu items, and then the columns are ingredients. And you can see for particular menu items, you require certain ingredients. And then on the right is shown kind of the McDonald's assembly line. This is how you might put together a Big Mac. I would like to think about how we create a lab to carry out the automated construction of what would come out of a Puppeteer, out of a cello, out of that planning. And so what's created at Boston University is something called the DAMP Lab, or the Design Automation Manufacturing and Prototyping Lab, that has a similar menu of items like the McDonald's, where we have individual operations or protocols that we might carry out, services, and those services require resources, and we can categorize and organize them. And the idea then is, instead of having the McDonald's assembly line, we have a very organized human assembly line that looks a lot like things that came from the biofab, for example, at the University of Washington, where we use software like Aquarium that was developed at the University of Washington and other people's software to organize our lab in a standardized way of doing things. What we'd also like to do is then also have highly automated processes happening in the DAMP lab, from work cells that are shown kind of on the left, which might look like something from the Global Biofoundries Alliance, that organization, or the Edinburgh Genome Foundry. We would like our own integrated kind of work cell. We have robotics in the lab, and what we're trying to do is automate the process of not only making the biology. I don't have time to get into all of this in this talk, but we also automate the manufacturing of those microfluidics. So the software not only goes to the robots and says, make this DNA, it goes to the desktop mills that are shown in the lower portion of this figure, that CNC mill thermoplastics to make 2-layer continuous flow microfluidics. So we can make both the hardware and the wetware in the same facility. And this is a dashboard. So the DAMP lab, the idea is at any given time, you can see all of the services that are being used. You can see the status of your job. You can see what else is going on. You can see it's kind of a social media platform as well. This is nice because the DAMP lab provides over 30 services as a core facility on the Charles River campus. So it's part of a larger effort that has been spun out of this career award. I mean, the career award had none of this vision in it, but it has led to, over the years, to this idea of a cloud lab that's built on the software that the career award really funded. So it's a mechanism to have that software have a real impact in the BU community and hopefully globally. And so people often ask what the DAMP lab can do. Just roughly speaking, it can kind of do things like, if we look at what can be done with our robotics, we can do 6,000 kind of transformations on a certain OT2 low-cost liquid handling platform, and as many as almost 13,000 cloning operations in a 24-hour span with some of the higher-end robotics. Julius alluded to a little bit of the COVID testing that I might talk about. I'll see if there's time in this talk, but part of what the DAMP lab converted to during the COVID-19 pandemic is the COVID testing facility for BU, where over 6,000 RT-qPCR tests are done every day in the building I'm sitting in right now. And it's using a lot of the automation hardware that we have obtained that was going to be in service for synthetic biology and eventually will roll back. And there's lots of lessons that we can learn from that. And on the right is what's shown for the micromill microfluidics. So putting all of this together, a lot of things to fit in here. Recently, I was awarded a Semi-Symbio award, and that kind of comes out of some of the road mapping effort through the Semiconductor Research Corporation, or SRC, NIST, and NSF, thinking about what techniques and ideas from semiconductors could be brought into synthetic biology. And so this is a collaboration with two folks here at BU, Wilson Wong and Mo Khalil. They make biological memory elements. Wilson does this with recombinases, where recombinase recognition sites invert or excise DNA and get it to become in certain states. So you can make DNA that physically changes itself to remember events. And then Mo Khalil works on kind of chromatin-based modifications that allow you to write or read DNA and put it into a particular state. And what I wanted to do then was build an ecosystem around that allows us to make these hybrid biological memories. And so if you looked at the devices from like a Wilson Wong, you could say there are many different ways to have inputs with this system, make them more sensitive, make them have more time. have different sensors for inducers, pH, temperature. Wouldn't it be neat then to take all of these different columns and combinatorially assemble these circuits in different ways? Well, that's exactly what the Living Computing Project would do. The Living Computing Project, the Expeditions Award, thinks about how to put these together using software tools and combinatorial assembly techniques. So then you can explore this space and make thousands of different designs. Well, that's great, but if you have thousands of different designs, how are you going to know which ones work? So the idea would be to make a microfluidic, and that microfluidic would be a droplet-based microfluidic that encapsulates those circuits in individual droplets. And then the microfluidics are designed and combined in different ways to do different tests. And then the idea would be like someone like Rabia, the professor here at BU, what she would do is then say, we're going to augment these microfluidics with electronics. So they're going to do kind of fluorescence or luminescence or other kind of electronic assays on chip. So you would put together a microfluidic using the components I previously showed using the CAD tools. You would make a microfluidic, a genetic circuit that you would send through this microfluidic circuit and have it measured its output. And then using microfluidic sorting, you could sort the cells, the devices into those that work versus those that don't, find the circuits that work well, and then put those circuits into a large, massively parallel screening system that's going to expose these circuits to different environmental characterizations and then determine which of these circuits work better versus others. And then all of this, again, this is very early stage, it was just awarded, but all of this was in service of a large scale kind of bioremediation platform where you could then deploy these microfluidics into the environment. And this is really great. I love what Julius talked about. He has a system, he's way ahead of me on this, where you might be able to go into the environment and think about measuring heavy metals. What's interesting about these systems is they already have electronics embedded, which include radios. So they can radio and relay their information as they're dispersed to a centralized location that can collate all the data. I'm cognizant of the time, so I'm going to say just a very brief statement about a couple other things NFS was involved in the last couple minutes here. Just briefly, SBIRs have been great. Part of this is you can't use any of this software long-term in a commercial setting. So Lattice Automation is a software company I spun out in 2013, which was supported by SBIR Phase 1 and Phase 2 awards to take much of the software I talked about and put it into a commercial setting in a variety of different areas. Works on lots of different places, agriscience, mammalian cell engineering, DNA synthesis, and it was recently put in IEEE Spectrum, just came out earlier last week about collaboration being done with GP Wright, an organization that is looking to think about how we engineer genomes to build their CAD platform. Again, all of this spun out from initial support that was bootstrapped from NSF SBIRs. A workshop called IWBDA helps to promote this. Every year since 2012, NSF has given some amount of money in the form of student travel awards to this workshop. And this workshop is really interesting because it has lots of interesting keynotes. It has abstracts that look like the abstracts you would submit to an electrical engineering conference. And it's been happening for lots of different years. It's spun out of a design automation conference. Then it became its own nonprofit effectively and has put on tons of different kind of workshops all over the globe. And so it's had over 1,200 participants, 197 presentations. You can see some of the stats there, but particularly interesting to me and from an NSF funding standpoint is it's funded almost 100 students directly to go to these conferences, which has been tremendously impactful. So that's been fantastic. And then I'll conclude with the fact that through the NSF Expeditions in Computing Award, we created something called STEM Pathways. STEM Pathways is a program that focuses on how we can take a look at underrepresented groups and women and get them in a pipeline at the undergraduate and high school and graduate stages to some extent to think about getting involved in research experiences, teaching them about how to navigate their academic career, get them involved in internships. It does lots of mentorship activities. We do outcome tracking. We track all of the students through this. So it's been a really great program. I love having undergrads work in my lab, but I hate the kind of wrangling and organization and mentoring process that often comes with doing this every time, every semester. You have to reinvent the wheel. This has created a stable program to really, I hate to say automate that because that's what I do, but it does automate much of this onboarding and has really been beneficial to the undergrads. Lots of projects, 70 some projects, hundreds of participants, has been really involved in iGEM, and we've been really successful there. So I'm going to conclude just with 3 questions. I know there's a lot, but the things I think about as I go forward in my career these days is I really am interested in what bio-readiness looks like. So a big part of COVID has woken us all up to, we don't want to, rebuild the wheel every time we have a biological emergency, and who knows what those will be in the future. So what roles do automation, data, computation, learning standards have in really making a national bio-readiness program? The other thing is, I think we need to do a better job of thinking about, as we transition from the benchtop to the laptop, we're doing things on a computer. What could we be learning? What could we be mining from that? How could we be using what people are doing computationally already to make biological design easier? And then finally, what is the commercial software landscape going to look like in 10 years? Are we going to have lots of cloud labs, biological services, none of the above, biofoundries? So things to think about. So thank you all for giving me a chance to speak. This has been a great, I'm looking forward to the workshop. So thank you all so much. As Julius alluded to and the other speakers, NSF has been a big part of my funding portfolio. And I thank you guys for all the support, and I look forward to the rest of the day. So thank you all.
Great, thank you, Doug. So what we'll do is we have a couple minutes for questions for Doug, and then we'll launch into a general panel discussion for about 10 minutes. So the first question is what are the challenges to having biologists or synthetic biologists use the design and automation tools that you are developing? From a distance, it appears that the barrier to entry might be high for adoption by classically trained biologists.
Yeah, absolutely. So one of the biggest challenges is bridging that gap between biologists and computer scientists. And so there are 2 parts. There's education on both sides. One is to educate the computer scientists about the biological realities to make sure the tools reflect that. And then I think there's a computational training that needs to occur in biological sciences a little bit. It's not enough just to know how to use email. It's going to be important for people to think about, everyone's sitting at a computer every day. So part of our challenge is lowering the barrier on the computer science side, making things approachable, making sure that people can express what they want. And then I think part of it will be changing some of the curriculum, the pedagogy, the training process on the biological side to bring people over. Some things that commercial companies can do is sit down and specifically foster those conversations through kind of contracts for service, consulting agreements and things. So I think there's a role that industry can play in bridging that gap as well.
Great, thank you. So the next question is for all of the panelists. Are there some grand challenge problems that the field of synthetic biology will prepare United States scientists to tackle? And what do we need to do to facilitate addressing those problems?
So Adrian, one thought about that question that I think was, we all alluded to in different ways as I'm sitting here thinking, is the need to sort of, not just generate a lot of large scale and rapid data, but really have ways where we can store it, where it can be centralized, where it can be cross-checked, where it can be shared quickly. I think the ability to sort of quickly share data and find mechanisms that we can, Doug was talking about automation, but automate it to search and to parse it and to really manipulate it in ways that are convenient and that lead to different purposes. It's something that I think synthetic biology and the rapid sort of development of a lot of the technologies is definitely posing that need even more, in my view.
And I'll add something to this idea of sharing is I think what's really going to be important is that we figure out a way to share. We figure out what form that should take formally. It's not going to be a bunch of prose emailed back and forth. Like, how is that actually stored? But then I think very importantly is how do we incentivize people to do this? Like a commercial company might they might think, what incentive do I have to share my knowledge? Like that's actually giving away my competitive advantage. And I think there's a way that certain things actually will end up making, like, this rising tide will lift all ships kind of analogy, that if we can figure out the right incentives and what people share when, it could end up being really advantageous. So us as a community, I think really could be thinking heavily about what the incentives to share could be.
I have a little bit of a different take. In my personal conviction is that synthetic biology stands to make the greatest impact in terms of sustainability across the globe. Amongst the many challenges facing us, these are some of the most serious, and they're also the ones highlighting a lot of the inequities in global society. And what I am inspired by synthetic biology is it is a biological solution. Biology is all around the globe, and it already is dealing with many of these challenges in interesting ways. And if we can figure out what those mechanisms are and how we can repurpose them, I think we can generate a lot of sustainable biotechnologies to address these challenges. I think it's going to take a lot of deep science and investment and changes in public perception, but I think this is kind of a path to becoming more one with the world than we currently are.
Yeah, and I guess I'll dovetail a little bit on that to say I think one of the other so the potential I think is enormous in lots of areas, and I think you've really seen that today. Like there's a huge range of possibilities where a lot of the core technology can be kind of rapidly redeployed at a new problem, which I think is really exciting. And I think some of the bottlenecks that I have seen are really some of the limitations are unexpected things, such as the fact that, for example, I've been really interested in cover crops. Maintaining our topsoil is a critical need, and this is a great opportunity. Cover crops are already a great solution to this, but they're not economically viable for farmers, even in the United States, and certainly in other parts of the world, it's even more of an issue. But part of the problem is we only pay farmers for a volume or a mass of crops, and it doesn't matter the quality of it at all. So like if you're trying to optimize something like nutrient value, or you're trying to optimize something like that, this is going to be higher quality feedstock for cattle, for example, because you've actually enriched the protein content, that actually doesn't show up in the economics at all. And so it's actually really challenging. So I think I have often found in synthetic biology approaches that they reveal for me, when you build things, it reveals what you don't know because things break or behave in ways that weren't expected. And I think that also actually ends up playing out in some of these other parts of the process where you see that the incentive structures, the values, are not there. And so they often don't incentivize the development of these products. So I think I just would echo, I think that connection is something, and I think something like NSF is extremely well placed to think about that really broad, integrated societal view, because I think that we're going to need to think across into the social sciences, into economics, to really actually make sure that we're incentivizing the solutions for things like sustainability or other practical applications. And once those things are in place or in conversation, then I think actually synthetic biology is extremely flexible and ready to actually meet those needs. And a lot of people are super excited. The people who I know who are going into synthetic biology, they want to be solving those problems. That is what drives and motivates them. And so I think that also will expand who's doing this work as well, because I think that also is, the people who are interested in intellectual puzzles and the people who are interested in making a difference in their community, it's often not the same people. And so I think we also will expand who's doing this work, which will then feed back into actually more creativity and different kinds of goals.
Great, thank you. So this next question is, again, for all four of you. But I just wanted to give a quick reminder that if anyone in the audience has questions for our panel, to please use the Q&A box down below. So the next question is, could you tell us about some of the key advances in synthetic biology that have enabled your research? And what are the key biological discoveries that have been important in your work?
So maybe I'll give a quick example of that, of something I alluded to in my talk. So I think that, years ago, even before I started my career, there was a lot of interest, like Julius, in designing sort of RNAs, designing synthetic RNAs. And one of these concepts has been really key for our very fundamental work, which really borrows this idea of a riboregulator. And it's a synthetic hairpin that essentially can open or close, and it has essentially has a controllable two-state sort of structure. And we've been able to use that for very fundamental studies, incorporating them in sort of, synthetic pipelines to inform us about how RNAs essentially do dynamic operations in vivo. So that's an example of translating something that was synthetically just constructed for a completely different reason that we've been able to capitalize and embed in other sort of synthetic constructs like fluorescent proteins and fluorescence assays, et cetera, and transcriptome technologies. And we've been able to discover new partners, new networks. So some of the discoveries that I talked about today essentially come from transplanting some of that technology.
If I can build off of what you said, Lydia, I mean, I think that's right on. And to broaden it a little bit, to me, one of the biggest innovations in the field is this discovery of design principles that act on many different levels. I think early days of synthetic biology was focused on this dream of harvesting parts from nature and putting them together like Legos and having everything work. And as we have all experienced, it doesn't really work like that. One of the fascinating questions I like to ask is, what parts do you use? Do you grab something from nature? Do you try to have a synthetic version of it? And I think there's a mix there, but what we've discovered as a field are these rules for making those choices so that things are predictable when they are put together in more complex designs. And that really enables things like computational modeling and prediction of behavior, going towards where Doug mentioned, which I think is where the field absolutely needs to go. CAD tools are an enabling tool for almost every other engineering discipline, and synthetic biology is just starting to get those. But I think it's the discovery of the design principles that's going to enable us to get there.
And I'll just say, I agree with the design principles. And then I'll also just add, I think it's also like the philosophy. So like for me, like I actually am relatively biologically agnostic in terms of what I mean, I'm not the cancer person or the bioremediation person. I'm always telling what's frustrating to my students, I'm interested in the process. And I think what I've seen over the couple of people over the past couple of years is other people also being interested in that process. That it's not enough that I just got this to work. It's that I documented that I put this somewhere, that I, shared this in a meaningful way that was outside of just my own local lab notebook. So there are biological, obviously scientific advances, but to me, a lot of it's just been that philosophy that part of my responsibility as a synthetic biologist is to contribute that back to the community.
I guess I would just add, on the technological advances, I think we wouldn't be anywhere really, in my world, at least, my part of synthetic biology, without the CRISPR-Cas system. I think that, and I think the Nobel Prize has done a pretty good job of identifying things. Also the evolution, like how we combine evolution into synthetic biology and how we use that to sort of optimize. So Francis Arnold's work and other things in that domain. So I think those are some of the ways that I think are also, they are both technology advancement, but they're also conceptual changes, real shifts in how we think about how to build surrogates. And especially I think this move from microbial systems into multicellular systems is actually a fairly significant hurdle still in the field. And I think that those are two of the things that are going to be really critical, in addition to some real changes in things like in vivo tracking systems, and being able to both decode and see what's happening, and also be able to then go back in and intervene in designed ways. And we always talk about it as semi-rational design. I think this is a phrase that I've come to really appreciate, because I think, especially in the multicellular context, it's usually, we start with the best hope, but I think we also have learned, and I think you heard that actually in Doug's talk too, like the advantages of being able, and in Julius's talk and in Lydia's talk, actually of looking at libraries and being able to sort of pull out what works best. And I think that's also partially an evolution inspired process, and then also being able to use evolution to actually optimize for ourselves these circuits. So those are just some ideas in that domain. It was a big question.
Great, thank you all. And with that, we are just about out of time. So that concludes our very first bioeconomy panel. I would just like to remind everyone that there is a PD meeting with our 4 panelists at 2 p.m. If you did not receive the link or you want us to resend it, please email Karen Cohn at kccohn@nsf.gov. And on behalf of the Bioeconomy Coordinating Committee, Bio and Engineering, I would like to thank all of our panelists and all of their introducers, as well as all of you for coming today. Thank you and have a great rest of your day.